Here at Economics, we're developing software that'll detect disease metabolites in the body fluids and speed up the whole process of diagnosis of urine or blood serum samples. Essentially, our software and instrument will be able to eliminate many of the steps involved in patient diagnosis and disease detection that is currently used in the medical industry. Right now, they collect a sample and then have to subdivide that sample up and ship it off to different clinics where they run individual tests. However, with our instrument and our software, the sample would only have to go to one location, and we'll be able to detect a variety of disease metabolites in just one individual sample.
The amount of data that's coming out of all sorts of different scientific tests now is overwhelming the researchers. The biggest thing that we're doing is automating the interpretation of that information and using a computer to obtain the information a lot faster than a researcher could. So for example, if a researcher would take an NMR spectrum of a biological fluid, they could generally identify maybe 40 compounds, and that would take about a day worth of work. Our software is at the point where it will identify and quantify 75 compounds, and it can do that in under a minute.
Certain compounds are indicative of particular diseases. So in producing our software, one of the essential components is to develop a database of different compounds. This becomes the foundation of our software in giving out a readout of the different compounds that are present in that sample. These compounds need to be tested in their purest form. So I will take a particular compound and, using different reference literature, I calculate what type of mass will be needed to produce a desired concentration. So once I've obtained the desired mass, I dissolve it in salt compounds, which simulates urine or blood. The process for testing the samples is fairly automated. I will run a variety of these compounds in their purest form through an NMR, which is a nuclear magnetic resonance instrument, which is basically a large magnet. It's encased by a jacket of helium and a jacket of nitrogen. These are all kept at very cold temperatures. We can basically press a button from the computer system and the sample is injected into the center of the magnet. Once a sample is injected into it, it'll give out a readout, which is called a spectrum. All the different compounds that are present in that sample will have a different signal. And from the different signals, you can determine what was present in that sample. And that's because we've developed an incredible database of all the compounds that could be present in biofluids. So on an ongoing basis, I am enlarging our database with new compounds and testing different urine samples or blood serum samples to determine what types of compounds are present. While I'm at my desk, I'm usually trying to find important references. I spend quite a bit of time on the biomed line. I also look up references to literature that can be found at the library. I also do quite a bit of work in Excel and spreadsheets. I need to confirm quantitation results, so that involves statistical analysis of results that are obtained. My research is either done over the internet or at the library or through scientific journals and literature that we do have here at the lab. This basically involves finding information about diseases and their associated metabolites that are found in body fluids. So, for instance, if someone were to have diabetes, you would detect a high concentration of glucose in their urine. And so those are the kind of things that I research. I also work with a software developing team. Because I'm constantly using the software that they produced, I run Spectrum through it to confirm that the results are accurate and that the interface is working optimally and there's no software bugs.
Why do you want to show it?
Occasionally, the software will have difficulties quantifying just one compound out of 75. And so from the statistical analysis that I do, I'll determine which compounds need a bit more work. One of my favorite pastimes is playing soccer. I play on a women's recreational team with 3 of my sisters. I also enjoy gardening, hiking, camping, and sewing. Chemistry is the foundation of everything. I always enjoyed chemistry. It was my favorite subject in high school. And what I do here is cutting-edge research. I find it intriguing to know our software can definitely change how the medical industry functions.